Welcome, everyone. For this deep dive, we're looking at some of the latest developments in cancer treatment.
That's right. We've got updates from several clinical trials and also some really intriguing preclinical work.
Exactly. We'll be pulling information from news reports on these trials, phase 1, phase 2, and some lab-based findings.
And the goal is really to distill the key insights, highlight what might be the next promising avenues in cancer therapy.
Okay, let's start with the clinical trials. There seems to be quite a bit happening in metastatic castration-resistant prostate cancer, or mCRPC.
Yes, definitely. First up, there's the COSMYC trial. It's a phase 1 study.
Okay, phase 1. And who is this trial for?
It's specifically for men with mCRPC whose cancer has already progressed after they've been treated with the newer hormone therapies.
You mean like abiraterone and enzalutamide?
Precisely, those second-gen androgen receptor inhibitors. So these are patients needing new options.
Right. And what's the treatment strategy in COSMYC? It sounds a bit complex.
It is quite interesting. It's a sequential strategy led by Dr. Samuel Denmead. It has two parts, or arms. The first is called BAT-ZEN.
DATZEN. Okay.
Yeah, it combined ZEN3694, that's a BAT inhibitor, with bipolar androgen therapy, or BAT.
BAT. So that involves high-dose testosterone. Seems counterintuitive for prostate cancer.
It does, but the idea behind BAT is that cycling these high testosterone levels might actually disrupt the cancer cells. And importantly, patients stay on their standard androgen deprivation therapy, ADT, throughout.
So the ZEN 3694, the BET inhibitor, what does that target?
It targets pathways involving the MYC protein, which often drives prostate cancer progression. So the hope is the combination of the BET inhibitor plus the hormonal shock from BAT makes the cancer more vulnerable.
Makes sense. And what happens after BAT-ZEN? You mentioned two parts.
Right. If the disease progresses while on BAT-ZEN, patients move to the second part, ZEN-ZA.
ZEN-ZA.
Here, they increase the dose of the ZEN3694 and add enzalutamide, another hormone therapy, while still continuing the ADT.
So sort of a 1-2 punch strategy. What are they primarily measuring?
The main goals are, first, to see if BAT-ZEN extends progression-free survival time without the cancer worsening based on scans or clinical signs. And second, to see if the ZEN-ZA sequence delays a rise in PSA levels, which is PSA, progression-free survival.
And secondary goals.
They're also looking at PSA response rates like a 50% or greater drop, actual tumor shrinkage, overall survival, quality of life, and of course, side effects.
When does this trial kick off?
Enrollment for about 50 men is expected to start around June 2025, and the trial is planned to run until June 2031, so a longer-term study.
Okay, moving on. There's another phase 2 trial, also an mCRPC combining FPI 2265 with Olaparib. What's the key takeaway here?
Well, a really notable point is that patients can receive Olaparib, which is a PIP inhibitor, without needing specific HRR gene mutations like BRCA.
Oh, that's significant. Usually PIRP inhibitors are restricted based on those mutations, right?
Exactly. This potentially broadens the patient population who could benefit. FPI 2265 itself is a targeted alpha therapy. It uses actinium-225 linked to a PSMA targeting molecule.
PSMAA targeted.
Like Pluvicto.
Similar target, yes, but this uses an alpha emitter, actinium-225, instead of a beta emitter, like lutetium-177 used in Pluvicto. Alpha particles deliver a more potent but shorter range radiation dose.
I see. And the trial design?
It's open label, multi-center with two parts. Part A is for patients who already had lutetium-177-based PSMAA therapy.
Like Pluvicto, as you mentioned.
Right. And Part B is for those who haven't had that type of radioligand therapy before.
So they're comparing effectiveness based on prior treatment history.
Essentially, yes.
Yeah.
They want to see if prior exposure matters. Within each part, there are two arms testing different doses and schedules of FPA 2265, always combined with Olaparib for the first 14 days of each cycle.
And the main objective?
It's really about finding the right dose and schedule the recommended regimen based on safety, tolerability, and initial signs of anti-tumor activity. Makes sense for a phase 2. Now, speaking of lutetium-177, we have phase 2 results comparing a treatment called FC705 against Pluvicto.
Yes, this is interesting. Both are lutetium-177-based radioligand therapies targeting PSMA for mCRPC. But the results for FC705 look quite promising in this head-to-head phase 2 data comparison.
How so? What did they find?
Well, for FC705, they reported a 60.0% response rate on PSMA-PET scans covering both soft tissue and bone metastasis.
60%. How does that compare?
Pluvicto's pivotal trial data reported a 29.8% response rate, specifically in soft tissue disease. So the FC705 number, covering both types of metastases, seems substantially higher.
That is a noticeable difference. What about PSA levels?
Also quite encouraging for FC705, 73.3% of patients on FC705 had their PSA levels drop by at least 50%.
And Pluvicto's reported rate?
There was 46.0% in his comparable studies. So again, FC705 appears potentially more effective by that measure too.
So better imaging response, better PSA response. What about safety? Any trade-offs?
The overall safety profiles were described as comparable. However, FC705 did show a slightly higher rate of serious adverse drug reactions, 10.0% versus 9.3% for Pluvicto. It's a small difference, but something noted.
And what are the next steps for FC705 based on these results?
They're moving forward. An INAND application, that's investigational new drug, has been submitted in South Korea to start phase 3 trials, and phase 2A trials have also begun in the U.S.
Okay, shifting gears slightly. There's a phase 1 trial enrolling for high-grade neuroendocrine tumors, NETs. This one involves an oncolytic virus.
That's right. This trial is happening at Sylvester Comprehensive Cancer Center. It's for patients with high-grade NETs from various places: lungs, GI tract, gynecological system, even prostate.
And the treatment?
It's a combination. They're using 2 checkpoint inhibitors, nivolumab and ipilimumab, together with an oncolytic virus called SVV001.
SVV001.
Seneca Valley virus. It's injected directly into the tumor.
How does that combination work, the virus and the immunotherapy?
It's a neat concept. The Seneca Valley virus is known to selectively infect and kill tumor cells. When the cells burst, they release tumor antigens and danger signals, kind of like putting up a red flag for the immune system.
Ah, so the virus basically unmasks the tumor to the immune system.
Exactly. And then the checkpoint inhibitors, nivolumab and ipilimumab, come in to boost that immune response. They release the brakes on the immune cells, allowing them to attack the cancer more effectively now that they see it.
Has this shown promise before?
Preclinical studies did show tumor shrinkage and durable responses in models. So this phase 1 trial is the first step in humans.
What are they hoping to find out in this phase 1?
Primarily safety, as always in phase 1, but also preliminary effectiveness. They'll enroll about 36 patients whose tumors haven't responded to prior treatments and compare outcomes to historical data.
Is there any way to predict who might respond best?
They're looking into that. There's a focus on a biomarker called TEM8. It's a protein on some tumor cells that SVVV001 binds to. So potentially TEM8 levels might help identify patients more likely to benefit.
Very interesting approach. Okay, one more clinical trial, update phase 1 results for a personalized cancer vaccine, PGD001.
Yes, this was tested across a range of cancers: non-small cell lung, head and neck, urothelial breast, multiple myeloma. Patients had already received standard treatments.
And it's personalized. How does that work?
PGSV001 is a multi-peptide vaccine. The peptides, small protein fragments, are designed based on the specific neoantigens found in that individual patient's tumor mutations.
Neoantigens, the unique markers on cancer cells.
Precisely. So it's tailor-made to train the patient's immune system against their own specific cancer.
And the phase 1 results, how'd it go?
Firstly, it was well tolerated, no serious side effects reported, which is always good news.
Definitely.
But the really striking part was the 5-year follow-up. Out of 13 patients, 6 were still alive after 5 years.
What?
And even more remarkably, 3 of those 6 patients remained completely tumor-free.
That's quite promising for a phase 1. Did they see immune responses?
Yes, they confirmed the vaccine-induced strong immune responses. It suggests the vaccine successfully activated the immune system against the cancer targets.
What are the implications then?
Well, it points towards the potential for these personalized vaccines to provide long-term benefit, possibly preventing recurrence by keeping the immune system vigilant. Of course, these are early days, small numbers.
Needs bigger trials, I assume.
Absolutely. Larger trials are needed, and also studies looking at combining these vaccines with other therapies, like checkpoint inhibitors, perhaps.
Okay, great overview of the clinical side.
Yeah.
Now let's switch to the preclinical findings. There's work on a molecule called SABER to boost vaccines.
Yes, SABER. It stands for Stimulator of Interferon Genes Agonist-Based ER Targeting Molecule. It's designed to make vaccines, both for cancer and infectious diseases, much more powerful.
ER targeting. What does that mean? How does it work?
It delivers the vaccine antigen, the bit that triggers the immune response, directly to a specific part of the immune cell called the endoplasmic reticulum, or ER. This happens inside dendritic cells, which are key antigen presenters.
And why target the ER specifically?
Targeting the ER seems to dramatically improve how the antigen is processed and then presented to C CD8 positive T cells.
The killer T cells.
The main ones, yes. So SABER basically supercharges the activation of these critical anti-cancer or antiviral T cells.
Supercharges. What kind of results did they see in the preclinical tests in mice?
They were pretty dramatic. For example, with melanoma tumor antigens, adding SABER boosted the CD8 T cell response up to 120 times compared to the antigen alone.
120 times. That's huge.
It is. It also performed much better than standard adjuvants, the traditional immune boosters in vaccines. In the melanoma model, it slowed tumor growth significantly, and 75% of the mice survived, compared to only 10% with standard treatment.
Any other cancer models?
Yes, in colorectal cancer models, they actually saw complete cures, and those cured mice were then protected against getting the same tumor again.
Wow. Did they test it against viruses, too?
They did, using a SARS-CoV-2 peptide, SABER, resulted in a 150-fold increase in T-cell activity and a 100-fold drop in viral levels for the Omicron variant. It also boosted antibody production with a different type of vaccine.
So potentially very broad applications. What's next for SABER?
The researchers are aiming for human studies, but first they need to do more safety checks and make sure they can produce it effectively. Scalability.
Makes sense. Okay, finally, there's preclinical work on a gene therapy delivery system called ENVLPE.
Right, ENVLPE. That stands for Engineered Nucleocytosolic Vehicles for Loading of Programmable Editors. Quite a mouthful.
Yeah. Yes. What's its purpose?
It's designed to deliver gene editing tools, like the popular CRISPR Cas9 system, into living cells more efficiently and safely.
We hear a lot about the promise of CRISPR, but delivery is often a hurdle, isn't it?
It absolutely is. Getting these complex molecular machines into the right cells without causing problems is a major challenge.
So how is ENVLPE different from existing methods, like using viruses, AAVs, or lipid nanoparticles, LNPs?
The key difference is that ENVLPE uses modified virus-like particles, but they're engineered to carry the fully assembled gene editor. This seems to avoid some major issues.
Like what?
Well, compared to AAVs or LNPs, ENVLPE appears less likely to trigger a strong immune reaction. It can also carry a larger genetic payload, and importantly, it delivers the entire editing tool ready to go, which might improve precision.
And did it work in preclinical tests?
Yes, they showed impressive results in a mouse model of inherited blindness. A single injection of ENVLPE carrying the CRISPR tool corrected the faulty gene and actually restored the mice's response to light.
That's amazing. Any other advantages?
Yes, they achieved this result using a dose that was more than 10 times lower than what's typically needed with other delivery systems. Higher potency means potentially lower doses and fewer side effects.
How might this apply to cancer, specifically something like prostate cancer?
The potential is huge. You can imagine using ENVLPE to deliver gene editors to solid tumors like prostate cancer to correct the specific mutations driving the cancer, or perhaps reprogram the tumor microenvironment to make it less hospitable for cancer cells.
Or even for cell therapies?
Exactly. Another exciting possibility is using ENVLPE to create universal T-cell therapies. You could edit donor T-cells to make them suitable for any patient, removing the need for personalized cell manufacturing each time.
So a really wide range of potential applications there.
Definitely. Still early, preclinical, but a very promising delivery technology.
Okay, so we've really covered a lot today, from clinical trials testing combination therapies, new radioligands, and oncolytic viruses, mainly in prostate cancer and NETs.
To personalized vaccines showing long-term promise, and then these preclinical breakthroughs with the SABER vaccine booster and the ENVLPE gene editing delivery system.
It's a real spectrum of innovation. And as we said, it's crucial to remember these are all at different stages. Preclinical work needs to translate to humans. Clinical results need confirmation in bigger studies.
Absolutely. Patience and rigorous testing are key.
But looking across all these different approaches, combination therapies, targeted radiation, viruses, vaccines, gene editing tools, delivery systems, it really highlights the multifaceted attack researchers are mounting against cancer. Thinking about all this, what area we discussed today do you find perhaps the most compelling or maybe holds a surprising amount of potential as we look ahead? It leaves you wondering which path will yield the next big leap forward.
Well, thank you so much for sharing your expertise with us today.
It was my pleasure.
I think our listeners will really appreciate this deep dive into the latest in cancer therapy.
Absolutely.
And for those of you listening, be sure to stay tuned for our next episode, where we'll be exploring another fascinating topic in the world of health and medicine. Until then, take care.